A gorilla named Kwibi was released into the forests of Gabon in 2004. He was young, raised by humans, and he went fully wild within months. Built a territory, joined a forest group, avoided people. 5 years passed. Then the man who raised him, Damien Aspinall, went back. He stood at the edge of the forest and called out. And a 400-pound silverback came out of the trees, pressed his face against Damien's, held his hand, and sat with him for hours. 5 years. No contact, no conditioning, no food rewards, nothing that any standard model of animal behavior can cleanly explain. So how does a wild animal living in a forest on another continent hold on to a specific human for half a decade? The answer sits at the intersection of neuroscience, stress biology, and a memory system so old that it predates our species by tens of millions of years. And once you understand how it works, you stop being surprised that animals remember. You start being surprised that we ever assumed they wouldn't. Let's start with what memory actually is inside an animal's brain, because most people picture it as a filing cabinet. You put an experience in, it sits there, you pull it out later. That's not how it works. Memory in mammals is a construction project. Every time an experience is encoded, the hippocampus, a small curved structure deep in the temporal lobe, coordinates a burst of activity across multiple brain regions. Sensory information, emotional tone, spatial context, and timing are all processed separately and then bound together into a single coherent representation. The stronger the emotional signal during encoding, the more tightly these elements are woven together. And the more tightly they're woven, the longer the memory lasts. This isn't a human invention. The hippocampus is one of the most evolutionarily conserved structures in the mammalian brain. Rats have one, dogs have one, elephants have one, whales have one. In 1957, a neurosurgeon named William Scoville removed the hippocampus from a patient known as HM to treat severe epilepsy. The seizures stopped. But HM could no longer form new long-term memories. He could remember his childhood. He could hold a conversation for about 30 seconds. But every new person he met, every meal he ate, every day he lived after the surgery was gone the moment his attention shifted. That single case study, published by Brenda Milner in 1957, proved that the hippocampus is not where memories are stored. It's the machine that builds them. Without it, the present evaporates. And every mammal on Earth has that same machine running. Now here's where the rescue part matters. Because the hippocampus doesn't treat all experiences equally, it prioritizes. And the single most powerful prioritization signal in the mammalian brain is stress. When an animal is trapped, injured, or dying, its body enters a neurochemical state that has no real equivalent in normal life. The adrenal glands flood the bloodstream with cortisol and adrenaline. Heart rate doubles. Blood pressure spikes. Breathing becomes shallow and rapid. In 2004, a study by Dominique de Kerven and colleagues at the University of Zurich showed that moderate levels of cortisol during an experience dramatically enhance memory consolidation. The stress hormones don't just make the animal feel afraid. They physically alter the way the hippocampus encodes information. Synaptic connections that would normally take hours or days to stabilize are locked into place within minutes. The brain, in a state of crisis, essentially writes in permanent ink. But there's a catch. De Quervain's research also showed that the relationship between stress and memory follows an inverted U-curve. A little stress improves memory. Moderate stress dramatically improves it. But extreme, prolonged stress actually starts to degrade the hippocampus. Cortisol in chronic doses is neurotoxic. It shrinks hippocampal neurons, reduces synaptic density, and impairs the very system that's supposed to be recording the experience. So an animal that has been trapped for days or injured for weeks is living in the worst possible window for memory formation. The stress is too high, too long, too corrosive. And then the rescue happens. Think about what that moment actually looks like from the inside of the animal's nervous system. Cortisol levels are at their peak. The hippocampus is running at maximum sensitivity. Every sensory channel is wide open, recording everything. The sound of footsteps, the smell of a specific human, the pressure of hands on the body, the precise moment the pain stops. And then the stress drops. Not gradually, it plummets. The trap opens. The net is cut. The wound is treated. And in that window, in the neurochemical space between maximum fear and sudden relief, the hippocampus does something extraordinary. It consolidates the entire experience, the threat, the rescuer, the transition from dying to not dying into a single, emotionally tagged, deeply encoded memory. James McGaugh at the University of California, Irvine, has spent over 4 decades studying this this mechanism. His work, published across dozens of papers since the 1960s, established that the amygdala, the brain's emotional processing center, modulates hippocampal consolidation during high arousal events. The more emotionally charged the moment, the stronger the amygdala's signal, and the more permanently the hippocampus records it. McGaw calls these flashbulb memories, moments of such intense emotional significance that the brain essentially photographs them. In humans, this is why you remember exactly where you were on the worst day of your life, but can't remember what you ate for lunch last Tuesday. The emotional weight of the experience determines its permanence. And here's what McGaw's research implies for animals. The amygdala hippocampal circuit is not a human circuit. It's a mammalian circuit. It exists in rats, in dogs, in primates, in elephants, in cetaceans. Every animal that has ever been rescued from a life-threatening situation has a brain that is specifically designed to record that moment with maximum fidelity and hold it for as long as the animal lives. But memory alone doesn't explain why the animal responds with what looks like affection years later. Plenty of memories are permanent but neutral. You remember your childhood address, but you don't feel a surge of emotion every time someone mentions it. So what makes rescue memories different? The answer appears to be oxytocin. In 2012, a team led by Teresa Romero at the University of Tokyo published research showing that dogs and their owners experience synchronized oxytocin surges during positive physical contact. When a dog looks at its owner, the owner's oxytocin levels rise. When the owner pets the dog's oxytocin rises. It's a feedback loop, the same one that bonds human mothers to their infants. But Romero's study showed something additional. The oxytocin response was strongest in dogs who had experienced high-stress situations with their owners. Dogs that had been through a crisis, a veterinary emergency, a frightening event, and then comforted by a specific human showed oxytocin spikes significantly higher than dogs in calm, routine interactions. The stress amplified the bond. This tracks with what we know about the neuroscience of pair bonding in prairie voles, which Larry Young at Emory University has studied extensively. In voles, the combination of stress hormones and oxytocin during a significant social encounter activates reward pathways in the nucleus accumbens, creating what is functionally an addiction to a specific individual. The vole doesn't just remember its partner, it craves its partner's presence. The memory isn't passive, it's motivational. And while we have to be careful about drawing direct lines from voles to gorillas, the underlying neurochemistry is remarkably similar across mammals. Oxytocin receptors are found in the hippocampus and amygdala of every mammalian species tested so far. The machinery for bonding through crisis is not species-specific. It's standard equipment. Now let's talk about what happens when you add time, because the question isn't just whether animals form rescue memories, it's whether those memories survive across years. In 2013, researchers at the University of Helsinki, led by Outi Vainio, studied long-term memory in domestic dogs using scent recognition tests. Dogs were presented with the scent of people they hadn't encountered in over 2 years. The dogs consistently showed recognition responses, elevated heart rate, increased sniffing duration, tail position changes. Specifically for humans they had positive associations with. Negative or neutral scents got a different response. The dogs weren't just remembering people, they were remembering how those people made them feel. Elephants push this even further. Cynthia Moss, who has been studying elephant populations in Amboseli, Kenya since 1972, has documented cases of individual elephants recognizing specific humans after gaps of more than a decade. In 2007, Karen McComb and colleagues at the University of Sussex published research showing that elephant matriarchs can distinguish the voices of over 100 individual elephants and retain those distinctions for years. The matriarch's hippocampus is doing something that most human brains would struggle with, maintaining a social database of over 100 individuals, each tagged with specific emotional and contextual associations across a lifespan of 60 to 70 years. If an elephant can hold the vocal signature of 100 family members for decades, holding the memory of one human who pulled it out of a mud pit is neurologically trivial. Bottlenose dolphins show similar capabilities. Jason Brook at the University of Chicago published a study in 2013 demonstrating that dolphins can recognize the signature whistles of individuals they haven't seen in over 20 years. 20 years. That is the longest social memory ever documented in a non-human animal. Each dolphin has a unique whistle, functionally a name, and other dolphins store that name and recall it with accuracy even after 2 decades of separation. When you hear about a dolphin responding to a former rescuer after years, the neuroscience doesn't struggle to explain it. The dolphin's brain was literally built for that kind of recall. And then there are the birds, which is where this gets uncomfortable for anyone who thinks memory sophistication requires a large brain. Corvids, crows, and ravens and jays have memory systems that rival primates despite having brains the size of a walnut. John Marsluff at the University of Washington published research in 2010 showing that American crows can remember a specific human face for at least 5 years and will respond differently to that face based on whether the associated experience was threatening or benign. 5 years from a bird. Marsluff's team wore specific masks while trapping and banding crows. Years later, crows that had never been personally trapped but had witnessed the trapping would still mob anyone wearing that mask. They didn't just remember, they taught their children to remember. The memory became cultural, a transgenerational grudge held by animals that weigh less than a pound. Now flip that. If a crow can hold a negative memory of a specific face for 5 years and pass it to its offspring, what happens when the experience is positive? The research is thinner here because it's harder to design controlled studies around gratitude than around threat. But Marsluff's own observations suggest that crows do differentiate between helpful and harmful humans, and that the positive associations, while less dramatic than the mobbing behavior, are also long-lasting. Crows have been documented leaving small objects, pebbles, bits of foil, shiny buttons near the homes of humans who regularly feed them. Whether that's reciprocity or something else is debated. But the underlying memory infrastructure is the same. The crow remembers who you are and what you did, and it remembers for years. But there's one more piece of the machinery that nobody talks about: sleep. In 2007, Matthew Walker and Robert Stickgold at Harvard published research showing that during REM sleep, the brain replays emotionally significant experiences at up to 20 times the speed of real time. The hippocampus fires in the same patterns it used during the original event, but faster, compressing the experience and redistributing it to the neocortex for long-term storage. This process is called memory reconsolidation, and it doesn't happen once. It happens every night. Every time the animal sleeps, the most emotionally weighted memories get replayed, strengthened, and woven more deeply into the brain's permanent architecture. Matthew Wilson at MIT confirmed in 2001 that rats replay the spatial memory of maze routes during sleep, reproducing the exact firing sequences their hippocampal neurons used while they were awake. The rats were literally dreaming about the maze. Now extend that to an animal that was rescued from a trap, a net, a river, a fire. Every night after the rescue, during every sleep cycle, the hippocampus is replaying that experience. The human's face, the human's hands, the moment the fear stopped. Not once, not twice, every night for weeks, months, potentially years. Each replay strengthens the trace. Each replay makes the memory harder to forget. The brain is not just recording the rescue, it is rehearsing it over and over, in the dark, while the animal sleeps. And this process is not optional. It's not something the animal chooses to do. It's automatic. Every mammal with a hippocampus does it. Every bird with a pallium does it. The rescue memory doesn't just persist because it was intense. It persists because the brain actively maintains it. And the real-world evidence for this is uncomfortably consistent. In 2003, a pod of bottlenose dolphins off the coast of New Zealand surrounded a group of 4 swimmers and herded them into a tight circle. The swimmers had no idea why until one of them looked down and saw a great white shark circling beneath them. The dolphins stayed for 40 minutes, slapping their tails on the surface to drive the shark away. The swimmers were rescued by a boat. But here's the part that matters. Rob Howes, one of the swimmers, reported that when he returned to the same stretch of ocean weeks later, a dolphin approached his boat, surfaced beside him, and vocalized directly at him before diving away. He couldn't prove it was one of the same dolphins, but the encounter happened in the same location, with the same approach behavior, in a species whose social memory spans decades. In 2001, a seal pup named Andre was found beached and emaciated near Cape Town, South Africa. A rescue team from the South African Foundation for the Conservation of Coastal Birds rehabilitated him over 6 weeks before releasing him into the ocean. 14 months later, a seal matching Andre's markings swam up to one of the volunteers during a routine coastal survey. survey. The seal approached from behind, nudged the back of her knee, and held position at the surface for several minutes while she stood in the shallows. The volunteer, Estelle van der Merwe, said she knew it was him before she turned around because he nudged the same spot she used to support him while feeding. So here's the picture that emerges when you put the neuroscience together. The mammalian and avian brain contains a memory system that was optimized by evolution for exactly the kind of experience a rescue represents. High stress, sudden relief, intense sensory input, a specific individual at the center of the event. The hippocampus records the experience. The amygdala tags it with emotional weight. Cortisol and adrenaline lock the encoding into long-term storage. Oxytocin binds the memory to a specific individual and charges it with positive social valence. And the resulting memory trace is stable across years, potentially across the lifetime of the animal. Karen Trendler, a wildlife rehabilitator in South Africa who has worked with injured animals for decades, has said something that aligns perfectly with the neuroscience. The animals who were closest to dying when they came in, the ones in the deepest crisis, are almost always the ones who respond most strongly to her later. Not because they're more grateful, because their brains were in the exact neurochemical state that produces the strongest, most permanent memories. The worse the crisis, the deeper the encoding. The brain doesn't choose to do this. It can't help it. It's running a system that evolved to make sure that anything associated with survival is never, ever forgotten. There's a lioness in Kenya named Kamunyak who, in 2002, did something that made international news. She adopted a baby oryx, a species that lions normally hunt. She guarded it, walked with it, slept beside it for 2 weeks. The oryx eventually died, taken by another lion while Kamunyak was drinking at a river. She went back to where the oryx had been, searched for it, and then adopted another oryx calf, and then another. 5 times over the course of a year, Kamunyak adopted the calves of her own prey species. Nobody has fully explained why, but the conservationists who observed her noted that Kamunyak had been found as a young cub, malnourished and alone, by Samburu herders who fed her and kept her alive before she returned to the wild. Whatever happened during those early weeks, whatever was encoded in her hippocampus during the transition from starving to safe, it appears to have altered the way she processed vulnerability in other animals for the rest of her life. That's not anthropomorphism. That's the amygdala hippocampal circuit doing exactly what it was designed to do, recording the conditions of survival and using that record to shape future behavior. The lioness didn't have a philosophy about mercy. She had a memory about being small and being saved, and that memory ran every time she encountered something small that needed saving. And here's what all of this means for the broader question. We spent a long time assuming that animal memory was simple, short-term, stimulus response, mechanical. The research of the last 30 years has systematically demolished that assumption. Animals form episodic memories. They tag those memories with emotion. They consolidate them using the same neurochemical systems that humans use. They retain them across years, and in some species, across decades. And the experiences most likely to produce permanent, emotionally charged, individually specific memories are exactly the experiences that a rescue involves. Maximum stress, sudden relief, a specific individual at the center. The reason animals remember their rescuers for years isn't loyalty. It isn't gratitude, at least not in a way we can measure. It's that the mammalian brain treats the moment between dying and not dying as the most important thing that has ever happened. And it records everything about that moment, every sound, every smell, every hand, with a precision and permanence that it reserves for almost nothing else. The brain was built to never forget the thing that saved it. And so it doesn't. Somewhere tonight in a sanctuary in Brazil, a jaguar named Jiquitaya is going to hear a specific voice outside her enclosure, and something in her hippocampus, something encoded during a week when she was a starving cub and a pair of human hands meant the difference between dying in a clearing and not dying, is going to fire. She won't analyze it. She won't weigh the evidence. She'll just walk toward the voice, the way she's done every time she's heard it for the last decade. Not because she decided to remember, because her brain decided for her a long time ago that this was something worth keeping.